Alrighty. Hello, everyone. It's morning where I'm at, but it might be afternoon or evening where you're at. So choose your time of day and hello. So we have a regular show today. Last weekend, we had a coffee chat because I was heckin' busy and couldn't sort out a topic fast enough. And so I thought we could catch up, talk about Halloween, which happened 2 days ago. It was fun. I wore a bunny onesie that is jammies with a hood that looks like a bunny, because I'm like, I just can't be bothered to do more than this because it was a very busy week and it's cold outside. Onesies all the way. You know what I mean? So, hey guys, hello. So I'm going to say hi to everyone in the chat. We're streaming on lots of different platforms now. I think DLive is new, so don't quite know how to navigate that. So it'll probably take me about a month to figure out how to do that new thing because it took about 2 months to sort out Discord. So that's how challenged I am with the newfangled technologies that the kids are using these days. So we're on DLive, Facebook, Twitch, YouTube, and Periscope. 5 different platforms. Good gravy, that's a lot. So catching up on stuff before we talk about today. Today, what we're going to talk about on the show here in a bit is essentially the latest science research that came out yesterday. Or the day or around Halloween. So like October 31st up through today. If you're not familiar with the National Science Foundation, they have a website called nsf.gov/discoveries. And you can go through and they'll post little miniature articles about what type of research they're funding and what kind of discoveries have come out of that. So I went to that website. I'm like, what's going on with science? How about we talk about that today? And so I pulled the studies that came out yesterday. We're going to talk about this today. So I thought that might be a good thing to do maybe every week or every other week or something, talk about what's new in science, because there's only so many topics that I can go over that's like considered, if you're talking about a fandom, this is canon, like cell biology, even though we have stuff that changes, evolves and things like that. It's just, there's the dogma associated with cell theory. I mean, just there's only so much that you can cover that's old. Well, I say old news, but it's new to people who have never heard it. But I have like hundreds of videos now, guys, freaking hundreds of videos about all kinds of stuff. And I can always probably get into more detail. But I don't know, it's like you can get like super hyper, focused on a thing. And then you start getting into terminology like the general population would not understand. And you would have to assume that they have a certain level of knowledge. So it's like prerequisites to get to the deeper information. And this is what I do here. It's like generalized science communication and science education, to where I go back over topics that the majority of the population doesn't have a working knowledge of. They might have been taught it before, but it's not something that's like in the front of their minds that they're thinking about. And we forget stuff sometimes, so we got to go back and do that. So I thought I'd say hi to people in the chat, and I'll talk about what we're doing tomorrow, and then we'll start talking about research. So we got Amelia, hello Amelia, and Mark Caesar, what's up? The Geek Room. Frank from the Geek Room is going to be on Oral Audacity tomorrow at noon. We've shifted the time to noon because Doc is an amazing human who works in immunotherapies, but he's 7 hours ahead of me. So when we were starting at 3, it was like 10 p.m. for him. So at midnight on what we call a school night, and he has to go to work the next day. So the Geek Room has been kind enough to switch spots with us. So we got Bull produces both of our shows. So that was a thing. And the Geek Room has been so kind to switch places with us. So Doc can continue to be on Oral Audacity. You know, he does stay relatively busy. So, you know, we might have a show every week. Sometimes it might be only a couple shows a month. But to me, Doc is integral to the show because he brings a lot of fun, and he's brilliant. He's just really, really smart. And his background in science, particularly when we start breaking down papers, I recognize I can hold my own. And he recognizes that too. Mel doesn't need my help. Yeah, okay. I know. I don't technically need his help to express science, but his perspective, he has different eyes than I do. And he has a different approach, and it's refreshing and it's fun. And he's Doc Savage. How can you not love to hear a guy talk about bad science, rip it apart in a Scottish accent? That's just joy right there. Am I right, Bull? That's just joy. What do you think?
Yeah, I like it. Works for me.
It's fun for me. So let me go back through. So Frank from the Geek Room will be on Oral Audacity tomorrow, and do check out their show. I'll put a link to it when I set up the next stream. We'll talk about what we'll be talking about here in a second. Let me finish saying hi to people. I need to finish saying hi to people. So we've got Danny Spitz. What's up, Danny? We've got XXXILD. Hello. We got Frustrated Atheist. What's up? Attorney in Bird Law has landed here in the chat. Fantastic. And we got Red Eye. So yeah, so we're all here. So tomorrow on Oral Audacity at noon Central, that would be 1 Eastern, noon Central, we will be talking about Dr. Spines. So just so you know, Dr. Spines, that's his Twitter handle. I've found some videos associated with him. Found some videos that he's posted. He's a chiropractor, and he has cancer coach alternative medicines on his website. And I've been coming through his website, and there's nothing that lists there about his cancer coaching, about what he does. So I called them today. I called them actually on Google Voice. I called them on Google Voice, and I talked to the receptionist. She was very nice. I didn't forget about XXILD. I thought I said that as exiled. Is it exiled? I think it's I didn't forget about her, Danny. So props. What's up? And so I called the receptionist. I didn't record the call. I could have, because they're in a one-party state. And I was like, this is just more informational. And so I got information, and I posted information on Twitter about it and tagging Dr. Spines. So if Dr. Spines wants to call in tomorrow, you know, he's welcome to join the chat. He can hit me up in DM, and we can bring him in, and he can defend his claims. But here's the thing. The receptionist told me that his wife, now late wife, she died, I believe, back in March, end of March. She had stage 4 breast cancer. The receptionist says and makes this claim, she didn't have any kind of medical treatment, no chemotherapy, nothing. She just did all the coffee enemas, and she was going, and she did the foot bath, and she did all of this other stuff that essentially are all the services that are on Dr. Spine's website, and it cured her. It cured her cancer. She had no cancer left. Stage 4 breast cancer. Now, if you're familiar with stage 4 breast cancer, the likelihood that anybody would ever survive from that is extremely low. You're at a higher risk for death if you go with alternative medicines over what we call medicine. There's a reason why it's alternative medicine. It's not actually medicine. It's other stuff. It's alternative, you know. it's not medicine. What do they say? They say if there's something in alternative medicine that works, they call it medicine. So, and I did. So last Sunday we were going to do this particular episode with on Dr. Spines, and I did contact Dr. Spines last week on Twitter about it to see if he wanted to join. And and he didn't respond. And I did post on some of his videos asking what the protocol was that his wife did on his YouTube. So I have contacted him in a couple places, so that's fine. It's up to him if he wants to talk or not. But I did dictate what his receptionist is saying. And essentially she says that's the draw that gets people to come in. They're curious about how his particular alternative therapies and how they cured his wife. So that's pretty low in my opinion. That's predatory. You're using your wife who died and she died in March from complications of surgery associated with a car accident. So she got in a car accident and her lungs apparently filled with fluid, and they tried to drain the fluid, and she ended up dying in surgery. But it wasn't the stage 4 cancer that killed her. They made a point to say that. The receptionist made a point to say that. And she said it's not on her death certificate. Well, if you die from a car crash, why would they list the fact that you had stage 4 cancer on there? That's not what a death certificate is for. So this will be an interesting show tomorrow. We're going to pick apart all of that nonsense. And if you're a patron, you already have access to our fake commercial for the show. Karen is on our fake commercial, Bull. Have you seen it?
She's back.
Karen is back. She's back, guys. Karen is making a comeback. She's got her own business, guys. And so she's our new fake sponsor for Oral Audacity. So she's got a brand new business she's a part of. So that'll be great. All right. So do check it out. Tomorrow, noon Central, Oral Audacity. We will be destroying the pseudoscience and the bad science of the death dealer known as Dr. Spines. So please do check that out. So, who did what now? Dr. Spines, check out you can find him on Twitter. He has a website. I'll be going through what essentially his cancer coaching is, according to his receptionist, who said his wife, who has stage 4 cancer, totally did not die of her cancer and was completely cured by his alternative medicine. So this is what she's telling patients and potential patients and people that call the clinic. I just called her today, actually. I called the clinic today. Yay. Hello, Immutable Destiny. Hello, Robin Eagleson. Frustrated atheist. Who's Karen? Oh, my goodness. How do you not know Karen? Karen is on Oral Audacity. She used to deliver the challenges for Doc on if it's a real product or not, if it's a real pseudoscience product or not. Yes, another quack who has their receptionist actually telling people that this quack cured, not killed, although that might be the case too, the cancer, probably. But this quack says he cured his wife's cancer and she died in March, April. So yeah, there you go. Hey, Tony. Not at all. Everybody in Discord. I'm learning Discord better, Tony. I need to add everyone. I need to add everyone on there. So 10 a.m. Central. Huh?
Actually, there's a bot. A bot should be doing that for you. Most people have a bot to do it.
Okay. I'll hit up Shane and ask him to do a bot for that for me. Because I know it does it when I upload a video, but I don't know if it does it when I'm live. So, okay. I'll hit up Shane in Discord and see if he can fix that for me. And Shane's very nice to do that. Discord confuses me too. All right. So let's jump onto what we're talking about today. We are talking about the latest in research. The latest in research. So the National Science Foundation has a website, nsf.gov/discoveries. So if you want to see what's coming out today with some of the research that the National Science Foundation actually funds, you can go nsf.gov/discoveries and see. So these particular things came out yesterday in Halloween. So this is brand new stuff right here, guys. Actually, this is all November 1st stuff. So these 4 things came out just yesterday. And the cool thing about the National Science Foundation is that they take the articles. So they kind of do what I do, or I kind of do what they do. You know, they existed before I did. So I kind of do similarly to what they do. I will say that. They take the articles and they just kind of break it down. So you can check out that stuff. So this is where I got my information. And I did go and check out the individual articles, and they link you. They link you to the actual paper. So they have a couple Nature publications in here. They have one on in ecology, and they also have one in it's to what is it, geophysical research letters. I think I forgot to put that one on my, I think I listed all of the ones and left off that particular one. So the last one on the Antarctic ice cliffs comes from geophysical research letters. It's a research letter. The rest of them are all actual studies from Nature and ecology. The bottom one is a research letter, and you can post letters. Sometimes it's still research, but it's mostly just an update on stuff that's happening. And that one has to do with marine ice cliff instability, and it's mitigated by slow removal of ice shelves. So we're going to talk a bit about that. But first, biomedical donuts, guys. That's a thing that exists. So biomedical donuts. Let's start with that one. So essentially, biomedical donuts, that sounds really cool. It's actually something that they call tori. So biomedical donuts, these are micro donuts that have been manufactured. Essentially what they do is they 3D print them, and they're coated with nickel and platinum. Now, this is actually a particularly cool thing because they notice that this stuff is kind of happening a bit with bacteria, and I'll get into a bit of more information with that. But essentially what they're trying to do is generate these 3D printed things, and they're coated with nickel and platinum. This could be a vehicle to deliver medicine, like targeted type of medicine, rather than when you take regular medicine, you'll get a shot, or you'll drink it, or you'll pop some aspirin, and then it has to go through and get processed in your system, and then it'll go and treat the thing. Using biomedical little micro donuts, that would be a targeted system where it would only affect the individual thing that it's trying to treat, rather than have a whole body effect. They made 2 different designs, and they're hoping that they can get these micro donuts to mimic some microorganisms. And this comes from this paper. The paper is entitled Shape Programmed 3D Printed Swimming Microtori for the Transport of Passive and Active Agents. So what does that mean? That just means they 3D printed things to get them to swim a little bit and mimic the behavior of microorganisms. And they're hoping to be able to have them be bioactive, to be able to engage with other biomaterials, whether it means interacting with proteins and maybe even signaling some cells. So let's talk about the article that was on the National Science Foundation. Let's dig in a bit. Okay, let's see. Let me catch up on the chat too. Is Amgen a thing? Not certain. NSF is not the same as NSFW. You know, this is the National Science Foundation, not the National Science Foundation working That's different. You're right. Immutable destiny. Oh God. Let's see. Yeah. You got like jelly juice and all kinds of stuff. Ooh. Yeah, it's amazing. Jelly's still around. How would the donut know when to stop doing its job? Well, um, okay, so that's a good question. So I used to work in nanoparticle technology. And essentially, what we were trying to do, we were using it for diagnostic purposes. So you can take various metals, you can coat them with variations of polyethylene glycol, and it's kind of like a sticky coating. So you can coat them with this little sticky coating, and you can attach antibodies to the outside. Antibodies essentially are proteins that can bind to different types of cells. So essentially, what you can do probably, and I'm kind of showing a practical application with these micro donuts, is that when you coat them, they coat them with nickel and platinum, and that kind of the reason why that's important is that it kind of bridges the gap between biological swimmers and like synthetic swimmers. And that's kind of important because if you're trying to mimic biological swimmers and interacting in a solution, trying to connect with other things, that's really important. But another key thing is, how are you going to get a cell to respond? And that's where you add receptors on the outside. So if you have a receptor, or if you have, you can have a particular molecule that fires off a receptor, another receptor, and starts the process of signaling. That's kind of what we use in medicine anyway, whether or not a receptor can be turned on or turned off to get a cell to behave a certain way. And I actually have a video on cellular signaling, so you can check that out if you really want to. Yeah, it's kind of like a finite thingy. It's a matter of whether or not it can turn a cell on or off when you're talking about cellular signals and receptors. Can I get this receptor to start its chain reaction of events in order to get it to behave a certain way? In the research that I did, I worked with the LPA2 receptor in computational chemistry. It's highly expressed in breast cancer cells. And so the reason why that particular cell has that high expression of the LPA2 receptor is because of the fact that cancer cells, the reason why they persist, number one, it's a failure for it to self-destruct. Okay, number one, your cells are not undergoing the self-destruct sequence. That is built into its DNA. We all have tumor suppressor genes. So if you have a self-destruct sequence is turned off, and it's turned off over and over and over again, the cancer cell is just going to continue to divide and make copies of itself. So that's the first thing. So number one, it's a failure of the cell to undergo self-destruct sequence. Number two, your other thing is your immune system. Your immune system will go and initiate that self-destruct sequence. The problem is this, that LPA2 receptor is highly expressed on more aggressive strains of epithelial cancers, including ones that are found in aggressive strains of breast cancer. And what that receptor does is when it's fired off, it prevents the cell from undergoing self-destruct sequence. So if you have a whole lot of those receptors, then you have a whole lot of, and it's fired off by lysophosphatidic acid, essentially LPA, hence the LPA receptor. So anytime LPA attaches to that receptor, it keeps that cell alive and says, nope, we're not going to self-destruct. Nope, we're not going to self-destruct. Nope, we're not going to self-destruct. So if you have like 10 of those or 5 of them, even half that, you have 5 of those on a breast cancer cell, they're constantly being fired off by LPA. They're constantly saving themselves from self-destructing. So an application, one thing that I did was try to find, I was generating a computer protocol with known antagonists, known things that'll bind to that LPA receptor, but won't turn it on, but it'll block it and keep it from being turned on. That's the key. So once you're able to block that receptor, then it can't constantly save itself, which reduces, it actually, it will likely increase the ability for standard medicines to work and take those cells out through chemotherapy and other forms of immunotherapies. So that's just kind of where we're going with that. So if you can use micro donuts, to block off that LPA2 receptor, that could increase the ability of standard cancer treatments to be more effective on more aggressive strains of cancer. So that could be one application of it. So this is all stuff that we can kind of do with that. So so it's kind of hard when you're 3D printing stuff, and this comes straight from Remy Baker out at Penn State. They studied this out at Penn State, and this was an NSF funded study. These microtori, and that's what they call them. They call them micro donuts. They call them microtori because they are active materials. This could be used to aid in micro mixing. So what does that mean? Baker says, we created two different designs, horizontal and vertical. Horizontal tori are printed on a flat supporting glass slide and then glazed with nickel and platinum. The vertical tori are 3D printed upright and dipped in nickel and platinum. Now, why is the nickel important and why is the platinum important? All right, well, 3D printing, you're dealing with plastic. Okay, you're dealing with different types of polymers. So the nickel, it helps for two things. Platinum does not stick to the plastic on the micro donuts, but nickel will, and platinum will stick to the nickel. And since nickel is magnetic, you can actually, in addition to this, manipulate the donuts with magnetic fields. So, cancer research brain working here. So if I'm able to 3D print this plastic thing, coat it in nickel, coat it in platinum, put some, things that could block off that LPA2 receptor, which would be kind of hard, thinking about it. But if I wanted to fire off or detect, I could use it in detection. You could, after getting a blood sample, detect whether or not a person has a particular cancer. That's what I did. I worked in cancer detection for exosomes, things that are even smaller than cells. Being able to detect something on an extremely small level and making it easier to find the cancer sooner, because detecting cancer, you have to have lots of cells there already. But if you can detect the exosomes of one cell that's coming out, that makes it a bit more sensitive test. So then if it has magnetic properties, after you go in and put these little microtori in your blood sample, they could then connect to, theoretically, if they had the appropriate antibody on the outside of them, they could connect to cancer cells or the like. Then you just stick a magnet on there. Clump it there, you could do that, and then just pull it right out and analyze. And if you're looking at trying to do genetic type of testing to see just how mutated a particular cell is, if you're looking at actual types of cancer cells, if they're able to work this out, you could coat one of these micro donuts in antibodies, detect the breast cancer cells, run a magnet over there, get it to stick on the side, slide it out, put it on, go and do some further testing, some even genetic testing if you wanted to extract some code from there. I mean, breast cancer cells do have DNA. That's kind of, so you can see. So there's different stuff, and it would just allow, additional research opportunities to be able to kind of dig in and see what's going on. So, there's lots of practical applications that can come from this. All right. Could the patient have MRI with those donuts in them? That's a good question. The thing is you already have a little bit of metal in your blood anyway. But if it's just a small, teeny tiny amount, I mean, you got to think about it. Your blood has iron in it. There's all kinds of different types of metals that might, platinum though. Platinum is not, I don't think platinum is magnetic right off. They might mess with the resolution of the MRI, but it also depends on how much is there and if you actually injected it into any particular portion.
They might, by virtue of the fact that they are metal, be detectable by the MRI and point you at something radiologically that you might not otherwise detect. Like you were talking about, it's great for detection. So if it's attached to something that you're trying to find, and you use an MRI to look through the whole body, for example, then you might see something that you wouldn't otherwise see on an MRI.
Yeah. It wouldn't be, well, see, that's the other side of this. With these biomedical donuts, we don't know how this would be in vivo. That would be in a living system. They would have to do mouse models probably. And then, if there was some kind of biomedical thing that people were able to do with these particular magnetic rings, that's essentially what they are, they would have to do different models with living things in order to determine if it's something that works. First, you'd have your cell studies, then you would have your animal studies. It takes a long time. It's probably something like that. If you're delivering medication, it takes longer than if you're doing detection. Delivering medication and seeing that it actually makes a difference takes a lot longer than just detecting if a thing is there. So for practical purposes, detection might be a more viable option, at least for the early stages of this type of biomedical tech. So, yeah. So how about, I'm catching up on the chat. Oh boy. Thanks, Nether. I'm just wondering how many would be in. Yep, ideally nanograms. Yeah, just few milligrams at most. Yeah, how a healthy body should be able to easily handle something like that. I would agree. I agree with that, Nether. All right. Let's talk about the next topic. We got 3 more scientific discoveries we got to get through. But yeah, biomedical donuts, man. Micro donuts. That would be cool. Yeah, heavy metal pre and post provocation lab test. That'd probably work. All right. So biomedical micro donuts are awesome. But what about magnetic eel memory, guys? Magnetic eel memory. This sounds like this should be in some kind of, use of like a James Bond, a kitschy James Bond film. Release the magnetic eels, But they use essentially they use like they're able to detect the magnetism in the tidal flows, like the magnetic fields in the tidal flows is amazing. So they have this internal compass. And this particular paper is called Glass Eels, and Gila. They imprint the magnetic direction of tidal currents from their juvenile estuaries. Doesn't that sounds terribly sciencey, but all they're doing is detecting the magnetic directions based on tidal currents from where they're born. This aids them in migration. It's uncertain if it's innate, if it's part of their genetic code, or if it's something that's imprinted while they're young. They're not clear on that, but they managed to test this in a magnetic lab. So let's dig in a bit and talk about this. So again, this is another NSF-funded study. And this research happened at the Miami Rosenstiel School of Marine and Atmospheric Science. Do check them out. They worked along with the Institute of Marine Research in Norway. And so there's this magnetic sense that they can imprint a memory of different types of water currents in their estuary where they were kind of reared as little baby eels and became little kid eels. Now this is particularly important because this is the first evidence of a fish species that uses this kind of internal magnetic compass in order to form a memory of which direction that the currents are going. So this is pretty important. So they essentially memorize the magnetic direction of the estuaries, and it helps them kind of get oriented where they are to where they can move and migrate. So they're able to go long distances. So essentially, this particular European eel, they cross the Atlantic Ocean two times in their life. First, after they hatch, which tends to happen in the Sargasso Sea, but they move as more than not as many, but more than 5,000 kilometers, and they coordinate with the Gulf Stream. Once they do that, they'll reach this continental slope right off Europe. Then they kind of they go through metamorphosis to where they become this transparent glass type of eel, and then they migrate across the continental shelf all the way down. Now these glass eels, they'll enter these estuaries, but they continue that migration upstream to freshwater later, until later in life. So almost is up to the age of 50. I think they can live as a little bit longer, but maybe after 50 they just kind of hang out. And as they're silver eels, they'll kind of navigate back to that Sargasso Sea where they were born. They'll spawn and then they die. So it's kind of like, yeah. So, but again, that's pretty cool. And how they tested it, they took these eels and they put them in their magnetic lab where they replicated the magnetic north, and it was rotated. And so essentially what happened is the glass eels oriented themselves towards the magnetic direction of the prevailing tidal current. And that was reoccurring, and that was occurring at their recruitment estuary, essentially where they were reared. So they use this magnetic compass they have in their head, and they'll orient themselves to the same current as where they were born. So it's pretty amazing. Golly. What do you think about that, Bull? Magnetic compass.
That's actually really cool. We use our eyes to do that for the most part. They wouldn't need sight. They could do this in the dark.
Yes, it is pretty cool. So they have like a magnetic compass in their head that's like, okay, they don't know if it is innate, if it is part of their DNA, or if this is something they just learned when they were growing in their little juvenile estuary. And so they'll be like, okay, this is the prevailing current in where I live. It's like a magnetic address. It's like their own form of magnetic GPS. That's just like the coolest freaking thing. But there you go. You guys are quiet in the chat. Are you still with me, guys? Oh, goodness bless. So, let's see. Another cool thing. Let's talk about the Galapagos and biodiversity. It is very cool. People are just, when I get my nerd on, man, I could just sit here and talk for hours, and we don't have that. We got like 14 minutes left of the show. Technically speaking, we can go a bit longer if we need to. You're listening. Yay. That's all I could ask for anybody, Robin. Just have a listen. Enjoy some science with me. That's what we do here. So, Galapagos and biodiversity. This particular paper is called Consumer Mobility Predicts Impacts of Herbivory Across an Environmental Stress Gradient. So, consumer mobility predicts impacts of herbivory across an environmental stress gradient. So, in brought down easy terminology, things that eat eventually foragers, things that eat like seaweed, they're able to predict the movement of these foragers, and they can accurately predict it based on the amount of environmental stress. And it's, how much stress is there. So what these particular scientists did was that they studied wave turbulence effect on sea animals. So they looked at the turbulence effect, from waves hitting these different populations. And what they saw is that the more diversity, and this makes sense, guys, basic genetics here, still being reinforced to this day. Basic genetics, evolution, natural selection, all of it in this lovely study. More diversity of an ecosystem equals resistance to rapid change, and particularly environmental change. What they found is that foraging rates drop with disappearance of species. So it affects all of that particular community. So this is useful in looking at whole community impact versus just an individual species. So while a lot of researchers will look at the individual species, you can look at the entire community now, particularly in these aquatic type of ecosystems, and kind of predict what the foraging rate will be based on the disappearance of singular species. But the more diversity you have, the higher the resistance to even the rapid change in climate. So let's talk a bit about these researchers. Let's dive in. Catch up on the chat. Quiet chat is a good sign. Awesome. Well, I guess so. I do like interacting with you guys, and I do read the chat. So Nether, doubt this is encoded in genes. Likely genes allow them to imprint, but the information wouldn't then be stored in genes. Life isn't just that, Lamarckian. No, you're true. There is an inherent need, like with golden retrievers, for them to go and get a thing. It doesn't matter. It's not like they have to go get the ball or go get the, it's not like they only get balls. There's a overwhelming need for them to go and get a thing. And, whether or not they bring it back, I guess that's what you got to teach them. So, yeah. Nice. Thanks for chiming in, Nether. And guys, I don't know everything. This is something, because I've returned to academia. As an aside, my job, my non-science communication stuff, because I do this for fun and I do coordinate. I'm growing a bit, particularly in the local area. I've joined a group of people who are science communicators, as well as scientists who work in various large research organizations here. And we're working in what's called Taste of Science. And what they do is they hold pop-up events where scientists can come and talk about their research to the general population. So I'm a part of that group now. I've been a part of Nerd Night. Now I'm a part of Taste of Science. So that's amazing. And I'm absolutely excited. I don't know everything. But I have returned to academia, and my job essentially, and where I work, there's a high amount of underprivileged individuals. And so what I do is I work to help them be successful in STEM academia and in their consecutive careers after that. So what I do is very rewarding, and I'm super excited to not only be able to help people be successful and help them be successful in their studies, but also I get to do cool stuff like this. But I even tell them, like, I don't know everything. Oh my gosh, I don't know everything. So that's okay. Hey, Angie Dell. Exiled expects me to know everything. Well, you learned something today. I don't. All right. So this Galapagos biodiversity thing. So I'm excited. I really am. So I do have to email the person back. They work at a world-renowned research institute here locally, and I've just been swamped. So I'm going to email them back today about being excited to be part of their committee to help them communicate all of the research that scientists do in this area. So it's amazing. We're just slow growth. It's still growth, guys, and we're growing the way that we want to here on the ScientistMel network and stuff. So it's exciting times. All right. So the Galapagos thing, that was funded by, again, the National Science Foundation. And this research was conducted at Brown University. And so essentially, these particular biologists studied the effects of wave turbulence on sea creatures. And it gives us a better idea of why biologically diverse communities are more likely to thrive even in the face of rapid climate change. And rapid changing environmental conditions. So John Whitman, who actually I think also took that picture of the Galapagos biodiversity, and the images that I have that are associated with my little breakdowns, those all came from the papers. So you can check that out. So credit for that. So this study was published in Ecology. And so it emphasized the impact of species mobility on their resilience. And so what they found is that highly mobile foragers, so those particular sea creatures that eat the things, eat the grassy stuff, appear to favor turbulent areas because these locations have a particularly abundant amount of food sources. And they found that the wave turbulence enhances algae growth, likely because of the fact that the water, the motion of the water promotes lots of delivery of nutrients. And so they were able to model this effect by analyzing the wave strength in conjunction with the foraging abilities of the various species of fish that they studied. And they were able to predict the extent to which these foraging rates would kind of drop off if different numbers of different species disappeared in the ecosystem. So ultimately, what's the thing here? Biodiversity, guys, is important for ecosystems, particularly where you have rapidly changing environmental conditions, i.e., climate change, global warming, and all of the different effects, ice caps melting, that sort of thing. So you have sea creatures being hammered in lots of different ways: temperature, acidity, wave turbulence. But the more diverse the species, the more that they can thrive. So that's kind of the takeaway of that. But that's evolution 101. And we just have more research to influence that. Thank you, science. It is pretty awesome. So the last thing that we're going to holler about here, Antarctic ice cliffs. Again, all of this stuff came out yesterday. So if you're wanting to see the cool stuff that scientists are doing and you're not getting it enough in your feed on your Facebook or on your Twitter, go to nsf.org/discovery. They fund so many different types of research, whether you're looking for biomedical science or ecology or climate change, or they had a study that came out a few days ago about face recognition software having gender issues. That's highly specific, isn't it, Bull?
Yeah, very, very specific.
It's highly specific. It's a diverse thing. So if you want to just kind of check out what's going on in various facets of science, I highly recommend going to that particular website. All right. So the next thing that I saw that I thought was pretty cool that came out yesterday. This is the one that was part of that research letter that I told you guys about. All right. So Antarctic ice cliffs. Now, the reason why this research was particularly important is because of the fact that they were really afraid that the Antarctic ice cliffs would break off and melt, and cause as much of a rise as 6 feet, which in the current water levels, which would flood areas like Boston. And so there's lots of concern about whether or not global warming, climate change, etc., is going to affect these particular ice cliffs. And how is it that, what can we predict is going to happen? Well, if you're not familiar with the ice cliffs, they span about 2 times the area of the US. And they're in the Antarctic. They're about 90 meters tall. So if you think of a meter stick is a yard, which is about 3 feet. Do a little bit of quick math there. 90 times 3 is what, 270 feet, something like that. So approximately around 250 to 270 feet tall if you're not metric yet. The rates of collapse have been overestimated. So according to this letter that was actually sent to, it was geophysical research letters. So you can find it in that particular journal. It's been overestimated. And what they found is that the ice shelves would need to break rapidly. It would have to happen right away, the supports associated with that, in order for it to break off and hit. So the rates of collapse that they assumed were these ice cliffs would essentially just fall into the sea. It's a bit overestimated because the breakage would have to occur rapidly. That's not happening. We're seeing very, very slow breakage over time. So let's dig in a bit and talk a bit more about this. So about 100 yards. So essentially, if you're trying to figure out how tall these are, essentially the height of the Statue of Liberty. So initially it was thought that this breakage, this rapid collapse under their own weight, because they thought that the ice sheet that's there, they need those cliffs those supports for those cliffs on this ice sheet. They thought that it would break off and could contribute to more than 3 feet of sea level rise by the end of this century. This would flood Boston and other types of coastal cities. Now, this particular NSF funded researchers at MIT, they found that these rates of collapse may be overestimated. So they published this paper in Geophysical Research Letters. It's still awaiting final review. It's been through peer review, so the information is accurate according to the peer review process. In order for this 90-meter ice shelf to collapse entirely, these ice shelves supporting the cliff would have to break apart rapidly within a matter of hours at a rate of ice loss that we have not yet observed in the current modern record. Now, if it were to take or melt away over a longer period of time, days or weeks, these particular scientists found that the remaining ice cliff wouldn't suddenly crack and collapse under its own weight, but it would just slowly flow out. And it would be kind of like, it would be a steady, slow flow, and it would be kind of like a cold increase. It'd be more like it's being released from a dam type of thing. So the worst case scenario. So Brent Minshew of MIT, who is a co-author of the paper, this is what he has to say. The current worst scenario of sea level rise from Antarctica is based on the idea that cliffs higher than 90 meters would fall catastrophically, like a doomsday film. But this is what he continues on to say. That scenario is probably not going to play out. Now, Paul Cutler, he's a program director at the National Science Foundation's Office of Polar Programs. They have a program just for that, polar programs. And Paul Cutler says, ice cliff failure has rightly become a focus of significant investigations by the glaciological community. Now, his particular polar program funded this study, and he continues to say, this project adds further key insights to that mix. So right now, research is moving to where we're losing our ice sheets. And so we need to understand how long is it going to take? How can we prepare? What's this going to have an effect on? Are those sea levels going to rise? What's that going to look like? So all of this research is particularly important in order to understand just how rapidly, if this dramatic breakage is going to happen or not. And right now we're not seeing anything like that. So that's good news, guys. No doomsday film about breakage of the ice sheet and the cliffs into the water, flooding coastal cities just yet. could still happen, but we're not seeing it right now. So there you go. So let's see. Question: is there legit scientific community studying of the effects of water modification, geoengineering on the current climate change issues? Nether chimed in to Robin's question. They're called physicists, meteorologists, and such. We know that short terraforming level projects projects nothing we can feasibly do will offset the amount of CO2 we release. Yes, that's true. Things are pretty bad right now. We need to lower our CO2 release, and perhaps maybe I think I've done a show on greenhouse gases. I know I've done a show on climate change. Perhaps I need to do a show on greenhouse gases, and why is it that CO2 is a greenhouse gas and not oxygen? I don't think people understand that structure and being able to respond to certain types of photons or the things that happen. And so perhaps that could be a topic that we could get into. What is a greenhouse gas, and what does it entail? So let's kind of catch up on some business. So that's all I got on latest research. But let's kind of wrap this up, and then I'll show you some of my sources. Essentially, yeah, and I left off the source on the Antarctic ice cliffs. But if you go to the nsf.org and you Google the paper that I said, you can check it out. So I got to fix those slides. And it's on geophysical research letters. Came out. It's still under, and I'll put it here. I'll put it in the chat here so you guys can gain access to it. Because, well, if you can.
In the description too later.
Yeah. So here, I'll just put these papers in the description later. How about that? And then you guys can do that. That's always a thing I can do. I'm also having website issues. I can't log into my own website right now, so I've had a hard time uploading slides. So apologies for that. I am working with my hosting group in motion, and we'll get that sorted out. So bring back the paper slides. Oh, Billy went back and watched some of the stuff on the Wayback Machine. I'm so glad you guys enjoyed this. So little bit of before we wrap up, I guess what are the 4 things we talked about? Let's do that. We talked about biomedical donuts, magnetic eel memory, Galapagos biodiversity, and all of the current information on Antarctic ice cliffs. So these studies came out just like yesterday. You can look at some of these sources. Here are my sources here. Also geophysical, the geophysical letters. I'll post these links. I'll post links to them in the description so you can check it out. So apologies to the researchers. Marine ice cliff instability mitigated by slow removal of ice shelves. That was Brent Minshew and Mark D. Ben out at MIT. So do check them out. You can find me everywhere on the internet. ScientistMel.com, patreon.com/ScientistMel. My patrons get early access to my fake commercials and all kinds of fun stuff. I'm on Periscope, YouTube, Facebook, Twitch, and now on DLive. So check that out. So let's get into a little bit of business here and catch up on the chat one more time. Okay, so here we go. I'm so glad you liked the show, Robin. I'm so glad you enjoyed it. So tomorrow at noon, we have Oral Audacity. Frank from the Geek Room will be joining us, and we will be talking about Dr. Spines, who is a chiropractor who claims that he cured his wife of cancer by having her do like 4 coffee enemas a day. He talks about that. I did actually call their clinic. So I called their clinic and talked to the receptionist, gave a little bit of information about that earlier. And she claims, she's telling cold callers who are calling his clinic that his wife was cured of stage 4 breast cancer by doing all of his alternative treatments. And so, but God, so I felt bad because his wife passed away in around the end of March. And they say it's due to the fact that she had a car accident and her lungs were filling up with fluid. And that was a side effect of the car accident, and she died on the table. Couldn't have any other complications associated with the breast cancer. I'm just saying. But I do have research that shows that people who turn to alternative medicine over, medicine are at a higher risk of dying from their cancer than people who actually get chemotherapy. And the receptionist said, essentially, she never got chemotherapy. She didn't get chemotherapy. She had no medical treatment whatsoever. He sells a lot of products, CBD oil, which is a derivative of, is basically oil from hemp. And so there are some claims that say that CBD oil, essentially THC, things associated with marijuana, there is THC related or actually cannabinoids. I've had a whole episode just on cannabinoids. Certain cannabinoids have some effects for different types of illnesses. But they're saying essentially that this CBD oil is the same thing as that. And it's not. CBD is not the same cannabinoid as others. And so they're misrepresenting actual science. And that's not what the papers say whatsoever. And we have a video on that. So, so Dr. Spines, I've invited Dr. Spines if he wants to come out. I have done it on Twitter. I've contacted him there, and I've posted on his videos on YouTube asking him for stuff, information. But he's more than welcome to come onto the show and support his claims, which I don't think he can do, particularly since I've done cancer research and Doc actually, world-renowned immunotherapy scientist. Nature did a feature on his work that he's done. Just as an aside, I've read the stuff that Doc has done, and it's amazing. And I'm very fortunate that I have such awesome friends in my life who, are not only smart, but they genuinely care about me, and they'll let me know when I'm being dumb. So guys who are my actual friends who truly care about me and my personal growth, thank you for letting me know when I'm being stupid. It means a lot to me. I do genuinely take your input to heart. You know who you are. And I'm so glad that I have you in my life. See, just as an aside, when you get criticism on the internet, you need to filter. The first thing that you need to do to filter, because I get criticism all the time. I don't always listen to it because the first thing I think about is, does this person care about my growth as a human being? Do they care about my personal growth as a human being? And if so, why? Why do they care? How does this, why do they care? So that's the first thing I think about when I go through. So it is a luxury having educators who know what they're talking about on YouTube. And so if you have a person in your life who genuinely cares about your growth as a human being, and they have shown it time and time again, that is the criticism to listen to, my friends. Those are the people you want to keep close. And I have friends tell me all the time that I'm being dumb. And I'm glad that they do that. And I know they care about me, so I take it seriously. Listen to who you listen to. Yes. Tatted philosophy. Billy calls me a derp all the time with technology. But not only does he call me a derp, because I am, with that, I own that. I absolutely own that. And I need to get better with Discord. And I'm trying. Like, Mel, you got to do this. Not only this, but he tells me how to be better. He actually helps me be better with the technology. So if you're going to dole out criticism and tell people stuff, at least be kind enough to tell them how they can improve as well, and do it in a kind fashion. That is constructive criticism, my friends, right there. Constructive criticism. Not only are you messing up, Mel, and that's okay. I mess up too. Here's what you can do to make it better. These are the conversations I have with Doc all the time, because I know the man is freaking brilliant. He's like, I dare say he's Einstein brilliant when it comes to immunology and, immune system stuff. He's like, so, and he has lots of life experience, but I know he genuinely cares about my growth. not only in STEM, but also as a person. So I listen to him, and I listen to people who have shown time and time again that they do actually want me to be a better human, and they express that to me. And they're like, "Well, this is my idea of how you can improve." That's constructive criticism. Can I get an amen?
No, I'm an atheist.
Fair enough. Fair enough. But there you go, you know. And so with that, guys, do check us out tomorrow at noon for Audacity with Frank from the Geek Room, hanging out with us, talking about the predatory death dealer known as Dr. Spines. You know, Tony says you can be an atheist and say amen, bull. We should fight about it for 4 days.
Nah, not worth it. Do you want me to say amen? I just did.
Make up all the good F in the chat for the derp. Billy, come on. Oh my goodness. So yes, you know, that's fine, you know. Is it religious appropriation to use the word amen if you're not religious? There you go. I've given you guys a topic to fight about for at least 6 months on the internet.
That's gotta be at least a 2-year topic. I feel it. I can feel it coming on.
Is it religious appropriation to use the term amen if you are not religious?
Remember, literally coined here, religious appropriation. Not cultural, but religious.
Religious appropriation. Oh. But we don't talk about religion on this channel. Not really. Only when religion and science overlap. We're science driven here. Oh my goodness. So there you go. So do check out Audacity tomorrow at noon. Frank from the Geek Room with me will be the incomparable Doc Savage as he scorns the simpletons in his wonderful Scottish accent. Makes all of the people swoon. All of the people swoon when he swears in his Scottish accent. So that is a show that's geared more for the adult crowd. You know, this is more family friendly for the most part. More, like teens and older, but the adult crowd is a bit more late teens and older. So there you go. And then go check out the Geek Room show. I appear on there sometimes and, have interesting things that have happened there, particularly when I lay down the smackdown on the bad science and the misrepresentation of science,
She's a glutton for punishment. She's gone there more than once.
We're like, here, Mel, come talk to this jerk on here who's saying things that are completely wrong. Okay, fantastic. And then I go and I pwn, PWN. I pwn. It's fun. All right. Well, anyway, we got a Mr. Rogers quote. Let's talk about a Mr. Rogers quote. Which quote? I can make Mr. Rogers is always relevant. Y'all just, hang in there. Let's see. Which section? Which section should we do? How about courage to be yourself? Let's pull something from there. That sounds good. Here we go. All righty. How about this? On confronting our feelings. All right, this comes from the book, The World According to Mr. Rogers. I always try to be like, what would Mr. Rogers do? That's what I do because Mr. Rogers is a role model of mine. I don't know how this man could exist in the current climate of things because sometimes you just get a little down. But think about what would Mr. Rogers do? And sometimes I fail at that. And I do my best to try to do what I can and say, okay, would I make Mr. Rogers proud right now? That's what I think. This is like genuine thoughts. You can ask people who like know me, and I'm like, I still have not watched the Mr. Rogers film because I can't do it. It just, I'll be a wreck for weeks. Anyway, here we go. Confronting our feelings and giving them appropriate expression always takes strength, not weakness. It takes strength to acknowledge our anger, and sometimes more strength yet to curb the aggressive urges anger may bring and to channel them into nonviolent outlets. It takes strength to face our sadness and to grieve and to let our grief and anger flow in tears when they need to. It takes strength to talk about our feelings and to reach out for help and comfort when we need it. Thank you, Mr. Rogers, guys. Address your feelings. It's not weakness, it's strength to be self-aware. Self-awareness is extremely important. It takes strength, not weakness, to do that and to curb those aggressive urges that you get when you want to be mean to somebody. So default to kindness. Think before you act and think before you speak. Kindness is free, and once you let that aggression out on somebody else, that's like busting a pillow full of feathers out into the world. You can get back most of those feathers. Can't take it all back, man. It's done. So think before you act, think before you speak, take a moment and curb those aggressive urges. I have emotions, guys. I get choked up sometimes. I got choked up. I did the science of Mr. Rogers and his communication and education style. How he teaches is actually actively studied by people, and that's their like PhD is on Mr. Rogers and how he educates. And I well up a bit. That is a thing that happens. I well up a bit on that. I do have feelings and emotions. I do get angry, and I'm a bit angry about Dr. Spines. And so that was probably going to come through tomorrow. So check that out. And I will see you guys soon. So I will see you guys next time. You always make each day special for me. You know how, by just your being you. Check us out tomorrow at noon on Oral Audacity. And then check out Frank on the Geek Room afterwards. I'll see you guys later. Bye.